electrodes and their characteristics. So in this topic, we will see what are the electrodes. See, till now, whatever we have discussed till now, we have seen what is the principle, how actually electric potential is generated inside our body. So what we call it as the action potential, how a cycle of action potential is triggered, how it is propagated, and what are the different types of electrical signals. We call it as bioelectric signals that we have discussed till now: ECG, UOG, ENG, EMG, and so on. Okay. Now, to measure these signals, we need some instruments. Okay. This instruments, it is actually again a separate branch in electronics engineering, biomedical instrumentation. Okay. Now, to measure the corresponding electrical signals, we need some electrodes. So these electrodes, they act as the interface between our body and the measuring instrument. Okay, so let us see. Now the electrodes, what we are going to discuss, these are called as biopotential electrodes because we are using these electrodes to measure the potential generated inside the body. That's why this term biological potentials, the electrodes used for measurement of biological potentials. So biopotential electrodes. Okay. Now we have seen earlier, actually the bioelectric potentials we are going to measure with our electrodes are generated in the body are ionic potentials, because last time we have seen different ion concentrations exist inside, because one there is some cellular membrane, and there are positive ions on one side and the negative ions on the other side, maybe within some fluid. Okay, so and we have seen last time these membranes, they have some semi permeable characteristics. Now because of this semi permeable characteristics, the movement of ions takes place from one place to the from one side of the membrane to the other side of the membrane, and that results in the current, which we call it as ionic current flow. Okay, now the potentials produced because of this, we call it as the bioelectric potentials. Okay, now see this, when we say bioelectric potentials, these are actually the potential existing inside our cells because of different concentration levels of the ions. Okay, but when we want to measure it, we are interested, or we can measure only the electronic potentials with the help of our electrodes. Okay, so by using these electrodes, actually the bioelectric potentials are converted to electronic potentials. Now we are going to see what are the corresponding principles of this. Okay, and this type of measurement, it should be noise free. See, particularly the amplitude level of these potentials is very small, as well as last time we have discussed, the frequency is very small, many times usually few hertz. Okay, and the measurement of low frequencies, it is actually more challenging. Okay, so that's why it is more susceptible to noise. So our electrodes as well as the measuring instruments or the circuits should be noise free, as well as the measurement what we get, it should be stable, because when we are carrying out the measurements, the body, there may be some physical movements in the body, right, because of the comfort level of the subject or the person. So in that case, the voltages may fluctuate, or there may be many other reasons, those we are going to discuss. So whatever potential we get, it should be stable. So we should have noise free and stable measuring devices. Okay, So the electrodes what we use many times, these are also called as recording electrodes. So these electrodes make a transfer from ionic conduction to the electronic conduction, because inside our body the potentials which are existing, those are ionic potentials. That is what we have discussed till now. So there are some ionic currents are flowing. So the function of these electrodes, it is actually transfer the ionic conduction to electronic conduction, because finally for the measurement purpose, for our electronic circuits we will need conduction in the form of electrons, not ions. So ionic conduction, it is inside the body, but for the measurement purpose, as far as the input given to the measuring instruments, they do not accept the current in the form of ions. Okay, so that's why the function of this recording electrodes will be to convert ionic conduction to electronic conduction. Okay, now these electrodes in actual applications we need for two purposes. The first purpose is for the measurement of physiological parameters by the impedance method. Okay, so because of the current variation, we calculate the impedance variation, and the impedance variation for different parts of tissues or of your body or the organs, it gives rise, because of the specific properties, the impedance it will be different corresponding to different types of tissues. Okay, so in one of the lecture last time we have discussed about this. Now the impedance, variation of the impedance of our body or particular type of organ or the cells, it conveys the information which is helpful for detecting any diseases for our doctors. Okay, now the other purpose of this electrodes is to stimulate irritable tissues in electrotherapy. So in case of electrotherapy, what we give, externally we apply some electrical signal to our body. Okay, you might have heard about this electrotherapy, or it is actually some shock treatment. It is just like a shock treatment. Now the intensity of the shock, it varies from different application to application. Now for this purpose, we'll have to, that means in this case we apply some external voltage to the body, and for that purpose also we will need this electrodes. Okay, now what are the types of electrodes? Now, depending upon different applications, the size of the electrodes or the characteristics of the electrodes are different. So, accordingly, there are mainly three types of electrodes that we are going to discuss. So, first one, it is called as the micro electrodes. So, these micro electrodes, as the name indicates, micro means a very small or the tiny electrode. So this is used to measure bioelectric potentials near or within a single cell. So we are going to measure the bioelectric potential of a single cell using this microelectrodes. It may be within this cell or it may be near to this cell. Okay. Then the second type of electrode, it is skin electrodes, skin surface electrodes. Now this type of electrodes, they are mounted on the skin surface. Okay, so the surface electrodes, they pick up the potential difference from the tissue surface when placed over it without damaging the live tissue. Okay, so while placing these electrodes, we have to take care that the tissues are not damaged. Okay, and this type of electrodes are in practice. This type of electrodes are used for measurement of ECG, EEG, EMG potentials from the surface of the screen. Then the third type of electrodes, it is called as the needle electrodes. So as the name indicates, these electrodes are like needles. So the needle is inserted in the live cell or in the particular tissue. Okay, so it is the deep-seated, these are the deep-seated electrodes indicate the electric potential difference arising inside the live tissue or a cell. So this needle, it is inserted inside the live tissue or the cell for the measurement of potential. Used to penetrate the skin to record EEG potentials from a local region of the brain or EMG potentials from a specific group of muscles. Okay. Now one word, what you see here, used to penetrate skin to record EEG potentials from a local region of the brain. So that means this needle is inserted in that inside the brain cell. So it is true. Okay, but and practically very little damage is takes place because of this. But it depends on applications. If the applications demand that the potential should be measured directly from the brain, then these needle electrodes are used. Okay. Now, how to select the type of electrodes? Okay. Now, the very important parameter, the electrodes should be compatible. Okay, because you are placing these electrodes on the body of the subject, right? So while carrying out these measurements, there should be some comfort level for the subject. Now, when I say subject, that means the person or animal, anything under observation or measurement. So in medical terminology, it is called as subject. So the subject should be compatible. The subject should not produce any artifacts. Okay. Now suppose for the measurement of EMG, suppose EMG means electromyograph. So this is related to our muscles. So now obviously during measurement of this myograph, you will need some motion of muscles, and when the movement of muscles is taking place, obviously there may be some problem in the in the contacts of these electrodes because it is connected to the body, and it may produce some disturbances. So these are called as artifacts. Okay, so the requirement is that the electrodes should not produce any artifacts. Then it should be convenient to use. Okay, so these are the choice of the electrodes. It should be compatible, should not produce any artifacts, and should be convenient to use. Okay, now as we have discussed, these electrodes are acting, these are actually electrode, it is the interface between the measuring instrument and the subject. Subject means, as just now we have discussed, the body. Okay, body of the person, human being, or maybe animal, like that. Now there are different layers during this interface. This side, this is the instrument. Okay, now output of the metal, output of the electrode, it is given to the instrument. So there will be instrument on the right hand side, then there is a electrode. Okay, and on the left hand side, this is a tissue or some body part. Okay, right? Now, this electrode, it is a metal part. When we say electrode, this is the metal part which should be connected to the corresponding cell or tissue, right? This is never connected directly. So instead of that, we use some medium here called as the electrolyte. Called as the electrolyte. Now, in chemistry, you might have seen when we measure potential with the help of these electrodes, it is always associated with some electrolyte, and that results in some half cell potential and so on. That I'm going to discuss shortly. Okay. So on the left side, it is the tissue or the body. Then electrolyte skin interface. This is the skin. So there is a and this side on the right hand side, it is the electrolyte. So it is electrolyte and skin interface, then electrolyte, then metal electrolyte interface, and finally the metal electrode given to the instrument. So these are different layers how this interfacing takes place. Okay. Now, when these electrodes are connected to the skin or the tissue, at that time, instead of connecting it directly, we use some lubricant type of thing called as the jelly. So this jelly, it acts actually the interface between the metal electrode and the tissue. And the electrolyte, it is included in this jelly. Okay. Now, on one side there is the electrode and the other side there is the electrolyte. Okay, so this forms some, it gives rise to some charge layer called as the double charge layer. Okay, and it is just like a PN junction diode. It works like a PN junction here. So the electrode tissue interface circuit involves transfer of electrons from the metal phase to an ionic carrier in the electrolyte. A charge double layer forms at the interface. So here it works like a capacitor here. Okay, because there is an insulator in between. On one side there is a positive charge, other side there is a negative charge. So it gives rise to some capacitance. Okay. So as you can see, the same thing here. On one side there is an electrode, other side there is an electrolyte. In chemistry, you might have seen this: cations, anions. That means the positively charged ions, anions, the negatively charged ions, A minus. Okay. So metal electrolyte surface, what we have discussed earlier. On one side there is a electrode, on other side there is a electrolyte. Okay, and this forms a charge layer. So some charge distribution exists at the electrolyte interface. So as I told you earlier, it works just similar to some PN junction. Okay, so as we have discussed earlier, this works like a capacitance also. Okay, and as there is a charge layer, it gives rise to some potential. So that potential we represent it by E, that is the potential, and the capacitance we indicate by C. The C represents the capacitance, and this R represents the resistivity of this material. Okay. So now based on this resistance, capacitance, and this potential, this will help us to draw to somebody. There is some disturbance from somebody's side. Please mute your mics. Dipali, your switch off your mic. Okay, so this representation of this resistance, capacitance, and this potential, this will help us to determine the equivalent circuit of this electrode, metal electrode interface. Okay, so this equivalent circuit that we are going to discuss next time. In chemistry, we might have seen related to the electrodes, there is some half cell potential. So a characteristics potential difference established by the electrode and its surrounding electrolyte depends on the metal, concentration of ions in a solution, and the temperature. So the potential developed at the electrode, it depends on three uh, three conditions: metal, concentration of the ions in the solution, and the temperature. So this decides the potential induced. Okay, so half cell potential cannot be measured without a second electrode. To measure this half cell potential, we always need two electrodes. Okay, usually hydrogen electrode, it is used as the reference, as the standard electrode. So hydrogen electrode, so the potential produced because of the hydrogen electrode, it is assumed as a reference, and that is assumed as a zero potential. So it is arbitrarily set to zero. So with reference to hydrogen, we measure the potential induced because of the other types of electrodes. Okay. Now this, why this half cell potential exists? Actually, this is related to the chemistry. So it is oxidation or reduction reaction, some chemical reaction taking place at the electrolyte interface, which leads to double charge layer, similar to which exist in electrically active biological cell membranes. Okay, so oxidation or the reduction reactions take place, and it gives rise to this half cell potential. And this diagram indicates how actually this half cell potential is used. Okay, so this is the hydrogen electrode here, which is acting as the reference, and this is the electrode material, and in between there is a electrolyte. Okay, so this meter indicates the potential because of this electrode material C with reference to the hydrogen electrode. And these are some of the half cell potentials because of different reference electrodes. Okay, now the potential because of hydrogen, it is assumed as zero. So all other potentials are with reference to hydrogen. So in earlier one of the slide for measurement of the ECG and so at the electric potentials, bioelectric potentials, we have seen we are using silver chloride, okay, as the electrode. So this is because it has a low junction potential. So silver chloride 0.222 volts and silver 0.780, and it is also stable. So the silver chloride potential, electrode potential, it is stable, and that is one of the reason why it is used for ECG electrodes. Okay. Okay, I think this part we'll discuss next time. So we'll stop at this point, half cell potentials. Now next time we'll discuss this polarization and all different types of electrodes, the classification what we have seen, the surface electrode, needle electrode, or the micro electrodes, and so on. So what are all different types of electrodes are there, how they look, what are the applications, working principle, what is the noise, artifacts, and so on. So those things we'll discuss in the next lecture. And I think in the next lecture, this may be the last lecture for this electrode and the unit number 1. So after that, we may start unit number 2. Okay, so let us stop for today here.